I am Leoni Bonamin from Brazil, and I work in Universidade Paulista in São Paulo, the city of São Paulo. And since 22 years ago, I study homeopathy as my main subject on research. And I'm veterinarian, and during all this time I work with experimental pathology. And now I'm learning in a post-graduation program about environmental pathology. And the homeopathy is inside the many different lines of research of the whole group. Homeopathy is my main line of research inside this context of environmental pathology. We are in a veterinary school, and in Brazil, but even in other countries, we realized that lots of diseases, mainly infectious diseases, are very related with environmental disturbances. For example, in Brazil, we had two important epidemics with veterinarian importance. For example, leishmaniasis and yellow fever. We realized that in the last few years, some diseases are related to changes in environment and environment conditions. We have changing in the weather, etc., and lots of proximity between cities and forests. In Brazil, it's very clear, this trouble. And new diseases are appearing. So we try to understand how homeopathy could help in these cases. We have two main experimental models we are working. The first one is to study in vitro models, in which some immune cells like macrophages are cultivated and challenged with different microorganisms, and we treat these cells with homeopathy and see what they do. And these findings can help to understand what homeopathy can do in clinical practice. Besides this line of research, there is another one more recent. There is mainly focused on environmental issues. We started to work with a little animal called Artemia salina. It's a micro crustacean. It's an invertebrate, so it's good to work because there is no bioethical issues regarding to these animals. They are very, very simple, and they are able to respond very well to homeopathic stimuli. And we had interesting results about. I'm going to talk about it in this conference. And I think with this model, we can understand another side of the problem and understand how homeopathy can help to find solutions regarding to the whole environment. Because if you see natural environments, there are plants, there are microorganisms, there are water, lakes, river, sea, trees, animals, and human beings. And the whole thing, the whole ecosystem can be treated or can be touched by homeopathy in a good sense, in a sense to recover the equilibrium, the lost equilibrium. And it's easily to see using experimental models in laboratory, and with Artemia salina we could see it very clearly. One way of working is using in vitro models with macrophages to study the effect of homeopathy on the immune system. And the other way of working is to study Artemia salina. And this little animal is able to respond very well to homeopathy. And there are very issues regarding to environment and this little animal, Artemia salina, mainly regarding to the sea contamination, for example. We can fix some things with homeopathy. About the crustacean, we are working with intoxication models. We intoxicate Artemia salina, and then we treat them with isotherapy. It's the same substance, but prepared according the homeopathic manner. And we could find out some interesting things. For example, Artemia salina intoxicated with glyphosate and treated with the isotherapy was able to change some aspects of their life cycle in order to protect the species from the intoxication. So maybe homeopathy could help to reduce the environmental impact of this kind of substance, or agrochemicals, for example, or even heavy metals like mercury. We did this experimentation too. We had similar results. About the potencies, we always use three potencies in the first essay: 60H, 30, and 200. It's always like this, in 3 levels of dilution. And after this, we can continue our studies using other potencies in between to know exactly if there is a curve where we can find the best potency for each case. But we always start from these 3, 6, 30, and 200 CH. And in this case of Artemia salina, we could see that glyphosate 6CH was the best potency. It was able to touch more parameters than the other potencies, with a number of animals towards 2,000 larvae of Artemia salina were studied. So we have a good number of animals, a good statistical approach, and I can be sure that 6CH is very good for this purpose. To protect Artemia salina is quite easy. Nature is able to do it. That's the most interesting, because people say, but what you say that homeopathy works, but what this thing you are showing, nature is able to do by itself. It's true. Nature is able to do it by itself, but homeopathy can facilitate this process. So to recover an equilibrium state of a certain population, for example, in a natural situation, we needed almost 10, 20 years, for example, after an environmental disaster, for example. Almost at least 20 years to recover the initial situation. But maybe with homeopathy, we can reduce this time for 10 or 5 years. And we had already some evidence about this time question. It's early to say something, to be sure that I asked, but we had some evidence that was very interesting. We have to pay attention to it and do more research to understand better, but I think it can be very useful in these situations. Everything is blind, so if we prepare the medicines and someone else changed the labels, and this code is locked somewhere, only this person is knows the codes, and so we treat with A, B, C, D, E, and up to the final, we never know which animals are being really treated or not. With Artemia salina treated with the isotherapy of glyphosate, for example, we could see that first there was less eclosion of cysts, so less larvae born after the treatment, but these larvae that were born were more healthy because the behavior showed more vitality, and the morphology showed a major proportion between healthy and defected larvae. And all of these parameters were measured according to quantitative methods and compared with very strict statistical analysis.